This presentation was recorded at the 2015 ANZICS ACCCN Annual Scientific Meeting.
Our last speaker, Steve Webb, has been a chairman of the Clinical Trials Group. He comes from Perth. I first heard him talking to us about ways to understand outcomes following intensive care. He's now interested in lots of stuff about how best to test hypotheses in a novel way. He's a big part of international collaborations, and he's going to tell us... What are you going to tell us, Steve? I forget.
I am going to talk on integrating research with clinical practice, and in particular about one particular trial design that I think is very much facilitates the opportunity to make research an integral and embedded component of routine clinical practice. So these are my disclosures, and I'll start again with a problem. Massive unmet need for better evidence in intensive care. This just gives you an idea of the proportion of treatments that we routinely provide to patients that are based on high quality evidence from randomised controlled trials. And it's not as though there's a shortage of patients to be enrolled in trials. There's, by one estimate, more than 5 million ventilated patients each year, and yet only a few tens of thousands are enrolled in a clinical trial. And all of the issues that Jack has talked about and the ones that Jean-Daniel has talked about are tractable and solvable if we can get many more patients into randomised controlled trials. So why trials are important? Experiment is without doubt the best judge of truth. Observational studies, even with the new the new approach to big data, offer a potential alternative. But the problem with observational studies is that they may or may not be confounded or have their confounding adequately adjusted by statistical means. And the problem is that you never really know whether or not the adjustment has been adequate. And so I think big data offers the prospect of giving us very small p-values and us being seduced by precision and not really taking into account whether or not confounding is present. And lastly, and I really do resent Jean-Louis' statement about all of our trials being negative. A lot of our trials, even in the mistermed use of the term negative, are not negative. We are frequently finding interventions that are harmful, and we're finding interventions that we didn't suspect to be harmful. They've gone into randomised controlled trials because people thought that they might be beneficial, and yet we get the surprise that they result in harm. And the only way that we discover from the amalgam of things that we do now that we could be doing in the future, which ones work, which ones don't work, and which ones are harmful, is to randomise people. And I also want to just introduce a term which will be familiar to many of you, which is comparative effectiveness research. We do an enormous number of things in intensive care without there necessarily being high quality evidence about their effectiveness. And that, when there are options about how we treat those patients, it drives variation in care. So what comparative effectiveness research is designed to do is to allow robust evaluation of those legitimate alternatives, the alternatives that are part of standard care. And I also think that, whilst this is an aside, that the ethical standards that are associated with clinical trials should be very much different for comparative effectiveness research, because these are treatments that are known or believed to be safe and effective. They are in widespread use. They are distributed already amongst the population, but it is not known which is the best treatment. And that's a very different ethical question to introducing a completely novel or experimental or treatment of unknown safety and effectiveness into a population. And so the way to move forward, in my opinion, is to replace random care with randomised care. At the moment, the ANZICS CTG is moderately successful. We're getting between 2 and 4% of prevalent patients into trials in intensive care units in Australia and New Zealand. So if that's what you can achieve, if success is achievable with 2 to 4%, I think we need to really think about what we could be achieving with 20 or 40 or 60 or 80 or 100% of patients being in trials. In the last 50 years, there's been massive improvement in outcome for childhood malignancies, largely as a consequence of the embedding of randomised controlled trials within routine care for childhood cancer. For decades, more than 90 to 95% of children with cancer around the world have been randomised in the centres that undertake these trials. And they invariably compare the best treatment that is known from previous trials with what is believed to be the regime most likely to result in better outcome. And so that has led to this lockstep improvement in mortality for a range of tractable diseases over a sustained period of time. But it's achievable because they are getting almost all of their patients into trials. They're not having random care, they're having randomised care. And so the solution, I think, is very much to embed research, and not just clinical trials, but observational research as well, in routine clinical care. We start with the presumption that we have patients that are going to need some form of treatment. We accept that there is substantial variation in that treatment, and rather than having which intensivist is on duty or how you felt that morning decide what treatments you're going to provide, we can just provide care in a way that's randomised rather than random. Additionally, we should utilise wherever possible the substantial amounts of data that exist within clinical management systems or within administrative data to reduce as much as possible the burden of data collection that's associated with trials. And the result of this is not so much a separate silo of research and a separate silo of clinical practice, but a merged single silo in which randomisation is a routine part of the healthcare system, and the funding for it comes from the healthcare system, not from research budgets. So a good place to sort of go off in a slightly different direction is to talk about some of the problems that exist with existing randomised controlled trials. I don't think there's anyone within the ANZICS Clinical Trials Group, notwithstanding our very substantial commitment to randomisation, who is not aware of an enormous number of problems that limit the way trials can be conducted, limit the way the results can be applied into practice and interpreted. So the first is that trials are costly, difficult, and frequently fail to recruit particularly effectively. And certainly part of this is because they're labour intensive, and they're labour intensive, as I alluded to earlier, because of these separate silos. It took about 6 years of recruitment to generate 1,600 patients to be recruited into the ARISE trial, and yet in that time there were thousands and thousands and thousands of patients coming through emergency departments, potentially eligible for the inclusion-exclusion criteria, but not randomised. The second problem with RCTs as we do them at the moment actually relates in some way to some of the things that Jack was talking about. On one hand, they're too broad, because what they do is they estimate the treatment effect for the average patient without taking into account the possibility of heterogeneity of treatment effect, or the potential impact of any other variables like comorbidity, or, as Jean-Daniel has indicated, the nature of the disease rather than the syndrome that's been responsible for making that patient eligible for that trial. So they're too broad, and we miss important bits of information that exist within the data because of that broadness. But at the same time, they can also be too narrow. They ask a narrow question, and perhaps a somewhat naive question, which is, in conjunction with this massive amalgam of other treatments that the patient is receiving at this time, if I just change one component of that treatment, will that make a difference to outcome? A much better question is, what is the optimal treatment for this disease? And that can be tackled by randomisation, but it needs to be tackled by randomisation that exists in a very different way than we think about it at the moment. Additionally, particularly from commercial trials, we see highly refined inclusion-exclusion criteria, because the purpose of a commercial trial is to generate evidence which is sufficient to obtain a licence to market a product. And so commercial trials are designed to have the highest likelihood of having a positive result, rather than generating the evidence that clinicians need, which is, what's the role of this treatment in the patients that I see on a day-to-day basis? Trials also take enormous lengths of time to complete. When ARISE started, I had hair. That's true. But we need answers to treat our patients now, not in 3 years' time or 6 years' time or 10 years' time, which is the length sometimes that it takes to do these trials. And I also think that this not knowing the answer for the trial for a sustained period of time contributes in some way to the element of discomfort that is real, that exists in association with randomisation for clinicians, for families, for ethics committees, that you just feel a little bit uncomfortable that the way that you get this answer, which will benefit future patients, involves experimentation with the patients that are under my care at the moment. And the last problem with trials is the one that Jean-Daniel also alluded to, that the results of trials are often ignored and not implemented, with even though there's good evidence that a particular treatment should be in routine practice, that it's not provided to a substantial proportion of patients. So is observational big data a candidate solution for this problem? As I said previously, I think the risk is that we'll get highly precise estimates of potentially biased treatment effect, and we are ultimately unable to tell whether or not the results of an observational study are valid, as many of them are, but unable to distinguish from those, the small minority, that are not valid. And so this really reflects what Donald Rumsfeld said when he said that the problem here is that there's a known unknown with respect to observational studies, and we just don't know whether or not bias has been adequately dealt with. So there are two new methods which are very much about to come to intensive cares in Australia and New Zealand through the ANZICS Clinical Trials Group that I think offer substantial prospects to be able to generate evidence, better evidence, and evidence more quickly, and more relevant evidence to our clinical practice. One is termed a cluster crossover trial, and I won't refer to it anymore except to briefly indicate that a cluster crossover trial involves randomising an entire intensive care unit to an intervention for a period of time, having a short washout period, and then crossing over to the other treatment condition. With that design, now funded through a grant from the HRC here in New Zealand, the PEPTIC study will compare H2 antagonists with PPIs for the prevention of stress ulcer prophylaxis. It will do that trial for $200,000, and yet it will have more power than any trial ever conducted by the ANZICS Clinical Trials Group previously. So that's a log improvement in efficiency associated with that design. What I am going to do for the rest of this presentation is talk about an entity called a platform trial. And I think a platform trial is a term that you will increasingly know about, a term that will be increasingly relevant to day-to-day practice in intensive care units throughout Australia and New Zealand. And I don't think we know yet if the platform trial is going to work or how it's going to play out, but if it does meet a substantial proportion of the promise that it offers, I think it's very likely that in 10 years' time we will be doing very few conventional RCTs, as you're familiar with, and that we will now largely in 10 years' time be doing platform trials. So this is a Viewpoint article that I'd draw your attention to, which was published in JAMA by Scott Berry, Roger Lewis and Jason Connor. And Scott and Don Berry, a father and son team, as well as Roger Lewis, are very, very closely involved in all of the aspects of the designs that I'm going to talk about today and the trial that we hope to bring to Australia and New Zealand in the not too distant future. What a platform trial is it's a piece of machinery, it's a bit of infrastructure, it's a system, and what it aims to do is to optimise the treatment of a particular disease as time goes by. It's not specific to any particular intervention, although it is specific to a category of patients. Jean-Daniel hates the word perpetual, but it's here. It's safe to say it. It's not safe to say perpetual in Europe, but it is safe in Australia and New Zealand. It's a perpetual trial that continuously updates the best treatment for all patients within the platform. It's not truly perpetual. Derek Angus, who's had a lot to do with developing this design, the question was put to him, when do you stop a platform trial? And the answer is you stop a platform trial when the problem is no longer a major issue of public health. But the idea is that it will roll for years, for decades, until diseases are solved. And we have worked out what optimal treatment is, and outcomes need to be no better. The first principle that's associated with platform trials is that they're highly adaptive. What adaptive means is, what adaptive doesn't mean is that you make up the rules as you go along through the trial. What adaptive means is that you plan the trial, the platform, in the beginning to deal with a range of potentially foreseeable situations, and then have contingent plans that are activated depending on the data that accrues during the trial. It allows it to navigate its own path depending on the results as they emerge. So rather than flying blind for 1,000 miles in a particular direction, stopping and seeing where you are, an adaptive trial allows you to fly for a certain distance, work out where you are, refine the direction in which you need to go, fly a little bit further, work out where you are, and refine the design as it goes. What you're used to, and what the ANZICS Clinical Trials Group has done up until now, are traditional so-called frequentist trials. And they make a whole range of potentially risky assumptions which can't be modified during the trial, even if you have access to the data to indicate that those assumptions are not valid. A first and critical component of all classic frequentist trials is a power calculation, in which one works out the plausible effect size and then an acceptable likelihood of failing to detect that difference to work out the appropriate sample size. Anyone who's designed trials or written grants for trials will know that where you actually start is at the budget. You work out how much money you can get, and then you work out whether or not the budget is sufficient for a treatment effect size that you think you can get away with in the grant, and that then determines your sample size. So in addition to estimating the right treatment effect, you make presumptions that you've got the right patient population, that you're actually studying the right treatment. And then you randomise on a fixed design until you achieve your sample size, and then you stop and you analyse. And there are really only 4 possible outcomes that can occur from a classically designed frequentist trial. The first of all is that the study reports no difference in outcome. But if it reports no difference in outcome, the interpretation is very different depending on whether or not the study turned out to be adequately powered, which is something that you only really know at the end of the trial. On the other hand, the trial might show difference, and then that leads to the situation that Jack described of, according to the JAMA guidelines for interpreting the medical literature, that if your patient has the inclusion criteria and none of the exclusion criteria, that treatment should be applied to your patients. It ignores the possibility of heterogeneity of treatment effect, and there are better ways of including the possibility of heterogeneity of treatment effect within trials. The last thing, and this is a point of substantial importance in terms of the ethical conduct of trials, is that in a classic frequentist trial, the result may well have been known well before the conclusion of the trial with sufficient statistical robustness if the data had been analysed. One of the perverse consequences of the way that frequentist statistics are designed are that there have to be very, very stringent rules for stopping a trial because of the possibility of type 1 errors. But if we think about the SAFE study, which compared saline and albumin, it would have been known with very substantial robustness well before the 7,000th patient had been randomised that for the vast majority of patients there was no difference in outcome between saline and albumin. It would have been known much earlier in the conduct of SAFE, if it had been analysed earlier, that albumin resulted in a 50% increase in mortality for patients with traumatic brain injury. So many patients continue to be randomised in SAFE, exposed to the harm that's known now to be associated with albumin. And lastly, within SAFE, there was a sepsis subgroup in which there was a strong signal towards benefit from albumin, but the trial stopped too early to know whether or not that was a real effect or not. In an adaptive trial, pre-specified rules would have allowed the trial to stop for most patients because of futility, to have stopped for harm in the group with traumatic brain injury, and have continued on to an appropriate sample size to evaluate the question in severe sepsis. So there are 4 key design features that are very, very different to any trial you've ever heard about that are associated with platform trials. The first is that they undertake frequent interim analyses using Bayesian statistics, which in brief allows the frequent analyses to be occurred without excessive risk of type 1 error. What that means is that the trial stops or the question stops when sufficient data has accrued, not when you hit a pre-specified sample size. The second feature is you test multiple interventions at the same time. That means that if you're testing 3 intervention or 3 domains of care at one time, you've already made this trial 3 times more efficient than a conventional trial. And this is also where the perpetuality bit comes in, because as soon as one domain has had an answer generated, a new domain, a new research question is pre-prepared and ready to be substituted into the trial. It can literally be that on a Friday the sites are told we are no longer testing intervention X, and on Monday, with ethical approval having already existed for some time, you're going to switch to intervention Y. The third component is termed response adaptive randomisation, and this makes trials very ethical, and I'll explain response adaptive randomisation in more detail in a couple of slides' time, because it takes a little bit to get your head around response adaptive randomisation. And the last is the use of predefined subgroups to allow for the evaluation of heterogeneity of treatment effect in the way that Jack has identified, allowing for the possibility that one group of patients benefits while another group of patients is harmed, which in a conventional trial will just give a null result. In a platform trial, those subgroups are specified in advance, randomisation is stratified by them, and it's possible using the interim Bayesian analyses to stop in one subgroup and declare and report and publish the outcome, whilst continuing the trial in another subgroup sufficient to accrue the information necessary to know the answer in that subgroup. It's a very flexible, very adaptive design that utilises the data that is accrued as a consequence of the trial to redesign itself according to pre-specified rules. So the first feature is these frequent Bayesian interim analyses in which accumulated data is used to update the probability that 2 treatments are either different or not different. In the trial that Jean-Daniel and I am involved in Europe, which utilises all of these principles, we stop for difference based on a 99% posterior probability that a treatment is superior, and we stop an arm for futility on the basis that there's a greater than 90% posterior probability that mortality is not different by more than 3%. And so we answer these questions in a rolling basis when sufficient data has accrued, and not by hitting a pre-specified sample size. We also test in this pneumonia trial 3 domains of care. We were randomised to choice of antibiotic, choice of immune modulation, and choice of ventilatory strategy. And all of those aspects will occur simultaneously, although not necessarily every patient will be eligible for every domain, and it's also not necessary for every intensive care unit that's participating to participate in every domain. If, for example, we're comparing choice of antibiotic, a patient that's allergic to beta-lactams will just not be randomised in that arm, but will still be randomised in the other domains of care. And as I've said, when an answer is generated, we roll on either to new, better factors that need to be tested within that domain, or completely different domains like fluid balance or sedation or other aspects of the general care that we provide to patients. response-adaptive randomisation. In all the trials that we've been involved in, there is a fixed proportion of patients that are randomised to each group. Literally a coin toss. 50% of patients have one intervention, 50% of patients have the comparator intervention. It's only convention that requires randomisation to be fixed one-to-one. There is statistical efficiency associated with one-to-one randomisation, and statistical efficiency drops off at different ratios, but not necessarily in a way that can harm the overall power of the study. What response-adaptive randomisation does is that the randomisation proportion changes throughout the trial on the basis of the accrued results at that time. During a trial, the statistical confidence with which one treatment is better than another treatment fluctuates, but it will over time have a progressive direction of trend associated with demonstrating if one treatment is better than the other, that is the case. In response-adaptive randomisation, the proportion of patients receiving the treatments that look like they are going to be beneficial increases progressively through the trial. Initially, when there's not that much confidence, it might be that 60% of patients get the better treatment. But as the trial progresses, 70, 80 or even 90% of the patients within the trial are actually randomised to the treatment that is going to turn out to be the best treatment. If the treatments differ in terms of mortality, what that means is that being in the platform lowers mortality for the participants in the trial. It's not that the patients in the future are getting the benefit from the trial, but it's that the patients within the platform are getting the benefit of the results that have already accrued that make a likelihood statement about what are the best treatments within the platform. So I think this is highly ethical, particularly in our discipline, where we enrol patients who are not conscious and not able to consent themselves. I think being able to say to patients when they've recovered and to families at the time that they are more likely to receive a beneficial treatment within the trial is a very substantial advance. And lastly, a platform evaluates predefined subgroups, allowing the questions to be answered in the subgroups that are most relevant and highly important to the talk that Jack presented so well at the beginning of the session. Bayesian trials have been around for a while. Berry consultants have run more than 500 trials, mainly for pharma, mainly for cancer, mainly for phase 2 over the last 20 years or so. But trials using Bayesian statistics are published regularly in the New England and in JAMA. They're accepted by the FDA in the United States and the EMA in Europe for licensing purposes. So I think we can have broad confidence in the validity of these methods. All of those four design features that I showed you previously have been used extensively in one or two of the components per trial. What's unique about the European trial that Jean-Daniel and I are involved in is this is the first trial that has incorporated all four of those components within a single trial. Nevertheless, there is still some statistical controversy about Bayesian analysis, and it is true that in a platform trial using this technique, that because if an arm stops early or because an arm stops because of accrued results, that it will, to a small degree, overestimate the treatment effect on a systematic basis. Not always, but sometimes. That's not important to clinicians because what you need to know is the qualitative answer that treatment A is better than treatment B. Where knowing the size of the treatment effect is important is in health economics, because it's only with a true estimate of the treatment effect that it's possible to determine cost effectiveness. I think that's a repeat. So these are examples of platform trials that are underway at the moment. I-SPY 2 is getting towards being a platform trial, but it's a trial of neoadjuvant chemotherapy, different chemotherapy and different molecular therapies for cancer in women with early stage breast cancer. And it utilises response adaptive randomisation and subgroups to try and match the best combination of treatment with molecular signature of the cancer. The trial that I've been talking about in pneumonia comes from the PREPARE consortium in Europe. It will enrol patients with severe community-acquired pneumonia. It's funded through a European Union grant, and an NHMRC grant application is pending. There are also platform trials under design for Alzheimer's, for glioblastoma multiforme, and many other diseases, all being run by Berry consultants from the United States. Have I still got a few minutes?
We've got to be finished by 10:15, so...
Right, okay. So I've run through the design features of a platform trial. A remap is what might be termed the big data variant of the platform trial. And this is where I really want to bring you back to the original issue, which is about embedding randomisation within routine patient care. So a remap is a randomised, embedded, multifactorial, adaptive platform trial. And it's about replacing random care with randomised care. Particularly where there's an electronic health record available, it can be used to screen and determine eligibility. It can be used to communicate allocation status as part of pre-specified electronic order sets, and it can be utilised to collect wherever possible in an automated way as much of the process and outcome data that we collect at the moment manually. But unlike observational big data, it retains random assignment, and it really does represent quite a unique fusion of continuous quality improvement. Because of the response adaptive randomisation, the best treatments are increasingly being allocated to patients within the platform. And because of its embedding and because of the use of response adaptive randomisation, it also overcomes one of the other major failures of conventional randomised controlled trials, because translation is embedded simultaneously in conjunction with the generation of new knowledge. Platform trials take a long time to get your head around them. They breach so many of the conventional rules that exist, but the rules that exist that we've been used to have existed as a consequence of the restrictions that are associated with frequenter statistics. I'm now giving this talk, but when I heard it, I had to listen to it about half a dozen times before I came to fully understand and appreciate the methods. It is a lot to take in in one go, but it is extremely likely that there will be a large number of ICUs in Australia and New Zealand participating in a platform trial before too long. Thanks, Tony.
Steve, I've got a question that's come in via the app. I'm going to alter it a little bit because it came at the beginning of your talk, but it sort of says that there's a perception out there among the public that being involved in a trial makes you a bit of a guinea pig.
Yep.
That you are taking this uncertainty, this randomisation risk, and that you might be getting a good treatment or the bad one. Now, the adrenal trial is maybe an easy trial to consent people into, because you can say the three key things you need in infection are source control, antibiotics, resuscitation, and we're giving you the best of all of that as we understand it. And we know that there's not a huge effect of corticosteroids because we've had some big trials done before, so we're looking for a small effect. And people like, we're going to fine-tune your treatment here, and by testing out in a lot of patients whether we can give you a little bit more help. And so that people don't mind that. But if we want to study a key thing, either with a frequentist trial or with the thing, and it might have a big effect, it creates a lot of anxiety. And how do we get around that and get people kind of to take the plunge? They have to trust us or trust the whole research process because they feel that they may be disadvantaging themselves by participating.
No, I mean, I consent people and I know exactly what you're talking about. So I think there are three things. The first is that I think we need to be substantially more honest with the broader community that so many of the things that we do are based on our best guess. and that at the moment we don't really know which ones are best. It comes back to that comparative effectiveness statement. 2 or more treatments that we are in widespread use that are believed or known to be safe and effective, but we don't know which one's best. So I think if you're explaining to families that we are testing treatments that are known to be safe or effective, but we just don't know which one's best, that can help. The second thing I'd say is that within a platform trial, being able to just simply say that this participation increases the likelihood that you'll get the best treatment can be very helpful in terms of overcoming those legitimate concerns and fears. Peter.
Steve, Peter Harrigan from Newcastle. What I wanted to ask is that if your ICU or your hospital is set up rolling on the platform trial, and that everybody is kind of, if you like, in the platform trial, getting what the platform trial does, and then the platform trial presupposes that somebody will have an idea for the next big thing.
Yep.
But the next big thing needs me to go out and try and have a little thought and say, why don't I give this a go in this N of 1 or 2,
yep.
and then somehow bring it to the team. But if I do that to the patients in the platform trial already, I pollute your platform trial with noisy treatments.
The platform's already polluted by all of the variation in the things that you do already, and that's not an issue.
So I can just have a go at something,
yep.
and if it looks good, I'll take it to the committee and say,
yep.
give this a crack. Okay.
But additionally, Peter, what has the capacity to exist within a platform is, let's say, there's an intervention for which there's uncertainty about its feasibility. I'm part of Carol Hodgson's team program of early mobilisation, and from a trial perspective there have been major issues about aspects of the feasibility, with concern that either in some places that early mobilisation can't be implemented, or the alternative possibility that when you do implement it in some patients, everyone goes, oh wow, this is amazing, and wants to implement it to all patients. And so in a trial perspective, we've had uncertainty about whether or not there's going to be sufficient separation to allow a trial to be conducted validly. What can be done within a platform is at a few sites to just run a feasibility trial within the platform seamlessly and just generate feasibility data, and we don't need separate funding for that.
Mary Lou Morritt from Sydney Children's Hospital in Randwick. Can you just tell me whether your perception is that NHMRC, IRB, ethics committees are mature enough to accept platform trials yet?
So I can't answer the question about the NHMRC; it's under embargo. But when we wrote the grant, we were very, very conscious that we were sending it to a conservative funding body, and there were major issues associated with being able to try and explain the method. But we really did go through a great many versions of the grant to try and communicate it as effectively as possible. You'll need to wait for the NHMRC results to know whether or not we've managed to sell it. IRBs are, I think, a major challenge. It's been approved by a single ethics committee at this stage at the lead site in Europe, and the questions that came back from the IRB were fabulous questions, and I think it went to a high quality IRB that was able to understand the design and the issues. Not all IRBs are of high quality, but I guess the challenge for us is to develop a package that allows these components to be disassembled and understood. One of the key issues, I think, will be, and certainly this is the way that we would have always intended to do it, we're only asking for ethical approval for the domains that exist at this time, and there'll be a fresh application for every time the treatments within the platform, for any new treatment added within the platform, because there's a unique set of ethical issues associated with each new research question.
Thanks, Steve.
There was a-
We've got more questions. There's several people who've got unanswered questions. Thanks for a talk that stimulated so much interest. They're going to have to be over coffee because we're right out of time. So we've got to go up there. I'd ask people who've got their questions left to catch Steve afterwards and thank him very much for a fantastic talk.